Couple quick announcements before we get started. First one is the biology seminar this week, tomorrow at noon over in SRTC. It's in room 101. Our speaker this week is Mark Slifka from OHSU, who does some really fascinating vaccine research. And some of you may have seen what I do on the side, so I'm particularly interested in that talk. Second thing, of course, is what's happening a week from Friday. First midterm, yes. I have now posted at least audio of all of the lectures on YouTube. There was a question I had right before class, is whether I could post MP3s as well as the MP4 video type thing. Would people be interested in that? Yes? Just audio as well as audio plus video? Okay, I will try and do that. The other thing is I'm going to try wandering around today just because I can get the technology to break again, maybe, so we can get all of that to work properly. But of course, before we do any of that, we really need to have our clicker questions. So today's first clicker question: two hybrid screens are a method to measure dissociation constants, rates of enzyme reaction, protein-protein interactions, the proteome, or protein-DNA interactions. So I'm glad to see that almost all of you agree with me. Unfortunately, it looks like we've got our answers here on the wrong screen, so I will now move them over so you can take a look at them too. The correct answer is C here, and 92% of you like that, which is good. I like that as well. So we can quickly select this answer and move on. Our next question is the transition from double to single-stranded DNA or RNA is called renaturation, denaturation, randomization, relaxation, or helicase. I hope that B is right. Yes, it is. So evidently, you were listening, or again, you had this in a previous class, and so you don't need to worry about it anymore. Our third clicker question today is the Tm of DNA increases with decreasing percent A plus T, increasing salt, the length of the DNA, A plus B, and A, B, and C. Okay, there seem to be some more differences of opinion on this one. So Tm of DNA increases with decreasing percent A plus T, increasing salt, length of the DNA, A and B, or A, B, and C. Nobody likes the length of DNA, which is good because that's not the case. And that's one of the really interesting things about DNA is that once you start to have denaturation, it happens really fast. And it's really independent of the length of the piece of DNA that you're looking at for the most part. Then increasing salt. Again, most of you seem to have gotten that from the lecture notes from last time. You found them quickly. But what does decreasing percent A plus T mean? Increasing percent G plus C. And so that means that we have which as the correct answer? D is the correct answer. So with that, let's move on and review a little bit about our proteins and DNA. And again, this is mostly for, excuse me, my purposes. You guys have any more questions, please ask me about them. So we talk a little bit about the protein-protein interaction assays, those being the two hybrid screens and the FRET assays, to some extent cross-linking as well. Talk a little bit about proteomics. All of these are protein-protein interaction assays, and the reasons that they're so important is that this is where you get understandings of what's really going on inside the cell, because these things are all interacting with each other. Talk a little bit about proteomics. Somebody asked me a question right before lecture. LC-MS-MS, the abbreviations in molecular biology are horrendous. LC is liquid chromatography, MS is mass spectrometry. And the key there is that you pick one peak in your first mass spectrum and then determine all the masses that are present in that one peak by breaking it down. So it's getting the individual ones. Yes? Yeah, so the question is, I'm just going to repeat it so it's on tape, is, how does that happen? Do you do one mass spectrum and then the second mass spectrum? No, you actually do them all online at the same time. And so as you're scanning across, and you program the mass spectrometer to do this, is it will pick out those peaks, and in each of those peaks do a second mass spec on it. So, but it's as you're running that, because you don't actually really know what peaks you're going to get, in theory, the first time around. And so it has to be something you're doing online. Other questions about the proteomics? We could have whole courses on proteomics here. Okay, DNA. Watson, Crick. Again, hopefully this is all review for you. Everybody read the paper? Yes? Nice short read. Good. Anybody really confused about it? I got a couple of questions on the discussion about, was it deoxyribofuranose? or desoxyribofuranose. The terminology has changed a little bit in the last 60 years, but the structure in and of itself, at least the model, is turned out to be extremely good. We've talked a little bit about major and minor grooves, and that's probably at least as far as I'm concerned, the most important aspect to come out of that structure, at least in terms of the structure, is that you've got really open space that you can read the genetic information that is between those two anti-parallel strands relative to each other. And again, we just talked about D and renaturation. Hopefully that's pretty clear to people. And then the last thing that we talked about was supercoiling. And I wanted to spend a little bit more time talking about supercoiling today, so I won't review it at this point. Any more questions about things other than supercoiling at this point? OK. Let's move up. That's the wrong direction. Don't need to do clickers again. We'll talk about, again, supercoiling. How do you get supercoiling? Or actually probably, well, actually, how do you get supercoiling? How do you deal with supercoiling? Because, as we mentioned before, one of the great things about supercoiling is that it takes a helix and can compact it, which is really what you need to do in terms of DNA. And again, one of the things that come out of Watson and Crick's model is that it's pretty inefficient. It's long and skinny. And that's not very useful in terms of packing things into small amounts of three-dimensional space. So supercoiling is absolutely critical for that process. How you do that is through the topoisomerases. And so we'll talk about the basically two different kinds of topoisomerases. You've got Topo Is and Topo IIs. And all that has to do with is Topo Is cut one strand of your DNA and Topo IIs cut two. RNA structure we'll look at a little bit. We can only just sort of very much scratch the surface in terms of RNA structures because sort of any nucleic acid structure you can imagine is found in RNAs. Then we'll talk a little bit about RNA processing. This is just messenger RNA processing. We'll talk a lot more about this later on in the class. Same thing is true for transcription. Translation we'll probably mostly end up covering on Friday, but we'll start a list a little bit with that today. I want to talk a little bit more about supercoiling. Here again, I mentioned the linking number, just the LK here. And all that the linking number is twist plus writhe. And twist is how many times the strands go around each other. And writhe is how many times those double strands cross. One of the things that I forgot to mention, or I guess hope that you all know, how many base pairs are there per turn in B-form DNA? There are 10 base pairs per each time you go around. And so if you know the number of base pairs in a particular molecule, 360 in this case, that'll give you exactly the twist. Just divide by 10. So here our twist is 36. In that case, in the middle, we have a writhe of 4. We've got 1, 2, 3, 4 crossing points here. And this is negatively supercoiled. That gives you a minus 4 writhes, so your linking number is 32. What that means is this molecule on the left-hand side, well, it's on left-hand side here, is equivalent to the one on the right-hand side. Because untwisting here, taking out 4 writhes, which is going to be equivalent to how many base pairs? 40, exactly. These two are topologically completely equivalent to each other. And this is one of the great things about negative supercoiling, or negative writhe. If you've got negative writhe, it helps untwist your DNA. Why do you need to untwist DNA? Replication, transcription, anytime you actually really have to have those hydrogen bonds available. So negative supercoiling is extremely useful, and of course that's what we mostly have in biology, is negative supercoiling that takes place. So this is really critical for compacting, but also helping out to open up those two strands relative to each other. Since it's so important, of course, you have enzymes that help do this. These are the topoisomerases. The topoisomerases change the linking number. So if you don't cut DNA, you're never going to change the linking number. However, as soon as you include a topoisomerase, you can. And what topoisomerases do, by cutting one strand, which are the Topo Is, or by cutting two strands in the case of the Topo IIs. One strand is going to change your twist, because it's just going to wrap around one strand, and cutting two strands and religating is going to change your writhe. But basically, they serve exactly the same purpose, because twist and writhe are interchangeable relative to each other. So the Topo Is, just diagrammed up at the top here, are going to cut a single strand, then pass that through one strand here, basically wrap it around again. Doesn't need ATP. You change your linking number by 1. The Topo II cuts two strands, pass those through each other. Topo II absolutely requires ATP. Turns out that Topo Is in many cases don't require ATP. It's not true in all cases. Changes by 2. Sometimes you might wonder, why do you need two different kinds of topoisomerases? Because they're equivalent, right? Twist you can change to writhe, writhe you can change to twist. Well, it turns out in some cases you actually have two DNA molecules that are linked to each other as a sort of chain linked relative to each other. This is what happens in replication in bacterial transcription. And so if these two are actually literally attached to each other, just because one ring is inside the other, you have to have a topoisomerase type 2 to pull those two apart. A topoisomerase type 1 is never going to be able to separate these two because it can just cut one strand. What do these topoisomerases do? Basically, all that they have are tyrosines in their active sites, which basically mimic the OH that you have on your backbone. So what they do is they'll attack the phosphodiester bond and basically exchange with the DNA which is there. So you form a phosphotyrosine here, and then this part of the DNA is released, and then you reverse that reaction at the end. These so-called tyrosine recombinases are extremely common in biology. We've actually been playing with some of them in my lab as well. There are lots of different kinds of topoisomerases, and the fact that there are so many different topoisomerases is a really good indication how important they are for functioning in biology. If you didn't need topoisomerases, you probably would have very few or almost none of them. In the case of these topoisomerases type 1, you've got 5 whole different families. Topoisomerase type 2, here you've got 4 different families. Lots and lots of different families of topoisomerases. What they all are is not critical, and I'm not going to expect you to remember all the different names here. I just did want to point out one of them, which is right here. This is the so-called reverse gyrase. Reverse gyrase introduces positive supercoils into DNA. Now, why do you want to put positive supercoils into DNA? Because that's going to be holding the two strands together, right? As opposed to the opposite direction with negative supercoiling, which is helping them come apart. Why would you want to do that? What if you're somebody as crazy as me and works with organisms that grow at 80 degrees Celsius? Those are always going to want to come apart. Think about the TM of DNA that we talked about last time. Those strands are always going to want to come apart. So positive supercoiling is actually going to help keep those strands together. And so it turns out that reverse gyrase here was originally discovered in organisms that grow at very high temperatures, 80 degrees Celsius and above. So it turns out that at these very high temperatures, you actually require this enzyme to keep those two strands together. If you look at, now, how do we know a lot of these things? Well, first was isolating the enzymes and looking at their activities, and then trying to figure out what happens to a particular piece of DNA. And I love this slide because basically it shows in the electron microscope exactly what's happening in the case of a supercoiled piece of DNA. And this is a classic kind of DNA, purified DNA out of a cell. This is a particular plasmid, small piece of DNA. You can see that it's supercoiled. It's wrapped around itself. If you put in Topo I, you end up with all of these relaxed circles, and all that's happened here is you've just nicked one of these strands, basically this pink strand right here, and because twist and writhe are interchangeable, this is going to go to the lowest energy conformation, which is this twist, which has given you, each of those 10 per turn, but no writhe. Again, sort of hopefully not beating the dead horse here. I mentioned catenanes down here at the bottom. If they are nicked, and nick just means you've got a cut in one of the two strands, then Topo I can separate those two relative to each other because it just needs that one strand to cut. If, however, they're not, then you can't have Topo I activity do that. If you have two single-stranded DNAs, Topo I can cut one of those, then it can interact with this other one, and then you can end up with double-stranded DNA. If you have these so-called knot structures, this is much more of a mathematical term, but it does turn out that you get a lot of these knotted structures in replication, in transcription, in just compacting the DNA. Remember, our DNA in one cell is a little over 6 feet long. It's got to get compacted down to about 10 microns. So it's not surprising a lot of these things get tangled up with each other. And so the biological way of dealing with these tangles is to have topoisomerases. How does Topo I work, or just a very general case of Topo I? They've got multiple different subunits, and basically one binds through that tyrosine residue that I showed you there that, again, is mimicking the phosphodiester backbone. And then the second part of the molecule basically binds to this opposite strand, pulls it through here. And the great thing about that tyrosine recombinase interaction is that tyrosine OH versus the OH in your phosphodiester are energetically extremely similar to each other. That's why you don't need ATP in most cases here to be able to reverse that reaction. So it's really a quite easily reversible reaction. On the other hand, Topo IIs do require ATP, and these are a much more complicated structure because they've got to bind to a double-stranded piece of DNA, bring these two double-stranded pieces of DNA together, cleave but hang on to both ends, and then pass the other end through in such a way that we've now recombined these two pieces relative to each other. Cut both strands, pass one double strand through, put them back together. Again, it's absolutely critical for resolving catenanes. These are double-stranded pieces of DNA that are linked to each other topologically but not covalently. And they're absolutely required for, again, most biology. It turns out if you knock out Topo IIs or Topo Is, all of them in any given cell, that cell is dead. So again, good indication for why you have them. Questions on supercoiling at this point before we move on and talk about RNA structures? Yeah?
Will the topos continue going until it's done, like all the way around the strand, or does it only do like one little?
So the question, I think, mostly has to do with processivity of topoisomerases. So once you've cut, how do you know to stop? And that's basically the question. And that's, in fact, a great question. And it's basically mostly seems to have to do with where the topoisomerase is relative to everything else in the cell, all the other proteins it's associated with. When we talk about replication, it turns out replication requires lots of topoisomerases. And so that's a way to look at it. But it's usually how they're associated with each other. But regulation of topoisomerases is a great question. And it turns out that it's kind of an aside, getting right to the end of the lecture today, but a lot of antibiotics work on topoisomerases. In fact, some of the best antibiotics out there work on the topoisomerases because they need these to be able to replicate. Okay, so let's talk a little bit about RNA structure. And again, if you're more interested in RNA structure, take Biochem, was it 491 or 492 with Dr. Lehman, because he knows way more about this stuff than I do. But basically, as I mentioned before, in terms of RNA structures, you can get pretty much any RNA structure you could imagine in terms of how these things associate with it. RNA has ribose instead of deoxyribose on the backbone. So that chemically, flexibly, pretty much the same as DNA. Uracil instead of thymidine, again, pretty similar, although the base pairing is a little bit different. But if you look at known RNA structures, you can have normal double strands, just like you have in Watson and Crick. And remember, in Watson and Crick, these are always opposite polarity relative to each other. So one strand's 5' to 3', the other is 3 to 5' relative to each other. So hairpins, double strands with big long loops. This is what's called a pseudoknot structure, where you have two different double-stranded Watson-Crick bases relative to each other. If you just draw them out in 2 dimensions, they look like this. In 3 dimensions, they're actually a more or less continuous Watson-Crick double helix with some extra loops that are hanging out on the outside. We'll talk about pseudoknots later on when we talk about transcriptional and translational regulation. Turns out they're critical for the virus replication, like HIV. But as well as having these continuous structures, you also have some discontinuous structures that you find all the time in RNA. Base pairing of 4 out of 5 of your nucleotides. Sometimes even less, where you have these bulge structures. You can have 3-stranded structures. You can have 4-stranded structures, etc. So basically, any possible way you can think of in terms of having RNAs interacting with each other, as long as the strands opposite each other are in opposite polarity, you're fine. And you can end up with lots and lots of different structures. Look at this pseudoknot in a little bit more detail, again, because it's important for virus. Of course, virus is the most important thing ever, but also for translational regulation, and it's not just a case of viruses. So the pseudoknot structure is down here. Basically, you have a stem loop structure, part of which can base pair to the RNA, which is slightly down or upstream from that. When I say downstream, I mean at the 3' end. Upstream, I mean at the 5' end. And so it's base pairing between the loop with that other piece of RNA, which is there. You can also have RNAs interacting through space, and it turns out this is really important for ribozymes. Those are RNAs which have enzymatic activity, and the most famous of those is the ribosome. Thank you. It's not even Monday morning. So it feels like a coffee machine in the back here or something. So yeah, all these interactions, 2 different pieces of RNA. These are just different names for them. You know, kissing hairpins or hairpin loop bulge contacts. Again, any way you can think of that these RNA molecules can be interacting with each other, and really important for having a particular structure, particularly a structure in 3 dimensions. Having structures in 3 dimensions is important for what? Structure is important for function, exactly. So try and get it out of you guys. So probably the best understood of the functional RNAs, at least in terms of activity, is the so-called Hammerhead ribozyme. What the Hammerhead ribozyme does is it cleaves RNA. It was originally found, of course, in viruses. But it turns out that there's a short piece of RNA which can have enzymatic activity, and that enzymatic activity is ribonuclease activity, cutting at a very specific position. And it's, I think, down to about 50 nucleotides. So it's a really, really small piece of RNA which can have really clear enzymatic activity, but the only way it can do that is by forming a very specific structure. And so that's the structure which you can see over here. Multiple Watson-Crick interactions, but also stem loop structures, etc., bulge loops, etc. And so these are those kinds of structures that you have. We'll talk about the ribosome now towards the end of today's lecture. First, I wanted to spend a couple of minutes, and again, this is sort of a teaser for what we'll talk about later on when we talk about transcriptional regulation. In terms of RNA synthesis is very similar to DNA synthesis. Again, it's phosphodiester backbone formation. The only real difference is you've got this funky extra OH here on your sugar in the backbone. You form a polymer by attaching the 3' OAH to 5' phosphate on the next nucleotide, which is coming in. It's a nucleotide triphosphate, just like you have in DNA. It's that hydrolysis of this high-energy bond here, which leaves you with beta-gamma phosphate, and then a second hydrolysis that happens between the beta and gamma bonds, which makes this reaction practically irreversible. We talked about the RNA polymerase, which does this, or the DNA-dependent RNA polymerase. We'll look at that in much more detail a little bit later on. One of the really big differences between RNA synthesis and DNA synthesis is RNA synthesis can start pretty much anywhere. And it is going to start pretty much anywhere, as opposed to replication, which has very defined places where it starts. So where transcription starts is dependent on a lot of other factors, and particularly the DNA sequences which are right around where you're going to make this copy. Transcription starts with what's called nucleotide number 1, also known as the +1 position. You have transcription from this position in a downstream direction. Now, these, of course, are double-stranded DNA, so you've got 5' and 3'. The downstream direction is going to be relative to the transcript, or the RNA, which is actually being made. Always starts at a 5' end, just like you have up here. 5' end here, which is then going to grow from 5' to 3'. And we'll see all polymerases that we know of go from 5' to 3'. There are no 3' to 5' polymerases that we've found yet. I doubt we are ever going to find any for reasons we may get into a little bit later on. So this is the RNA which is made, but of course you've got these double strands. There's the so-called non-template strand. That's the one which is basically pulled out of the way by the RNA polymerase. And then the template strand, that's what you end up copying. It's the blue strand that we have over here on the left-hand side of the slide. This place that you find is, again, where you have to start transcription. First thing you need to do is pull those two strands apart. Then you've got to start with the original nucleotide. How is this found in the first place? Was due to these DNA sequences, which are usually right upstream, and so not where you're getting the RNA transcript being made, known as the promoter sequences. And we'll talk a lot more about promoter sequences as we move along here. The RNA polymerase itself here is a blob. We saw the structure, I think in lecture 2 or 3. Really 3 main stages that happen here. You've got to start, you've got to go, you've got to stop. So you all think of this as the green light, yellow light, red light, except here the orange light is or the yellow light is the one which stays on the longest. Initiation also really has 3 steps. First, you have binding of your RNA polymerase to that specific sequence, again, the promoter sequence. Once you have binding to the promoter, then you have what's called open complex formation. You pull the 2 strands apart because, unlike just reading the DNA sequence, here you have to be putting in hydrogen-bonded nucleotides to that template strand. So pull those 2 strands apart. It's the open complex formation. And then the RNA polymerase will put in a nucleotide at that first position. This is your initiation step. Once you've put in these first one, or usually a couple of nucleotides, then you move on to the elongation step. And this is just adding nucleotides one after the other. This elongation step is the really easy one. It's the initiation one which is really hard. And it turns out that there are a lot of changes that happen between initiation and elongation. And one of them, which is a concept we'll talk about a lot more again later, particularly in terms of DNA replication, is something called processivity. And processivity means that you just stay on your template for long periods of time. And RNA polymerases are great at this. They can stay on for literally millions of bases at a time just with the RNA polymerase itself. And so that's a little different relative to DNA polymerization, which again we'll talk about a little later on. If you're highly processive, at some point you do have to come off of that template. And so now you also have the termination step, which happens down here at the bottom. And we'll talk about how each of these stages actually take place. Again, we talk about transcription or regulation later on. Each of these three steps is regulated. Most of the regulation seems to be happening at this first step, initiation, getting the RNA polymerase to the promoter. But there's certainly regulation that happens at later stages. What are those structures of the genes that we've talked about? Here, there are really two very different kinds of gene organizations that you have. And it says here prokaryotes. This should really be bacteria. 99% of the time when you see prokaryotes in a textbook, it means bacteria. They're archaea, but they're kind of in between here. And eukaryotes, which we have over here on the other side. In bacteria, almost all the time you have multiple genes that are all right together, which are being transcribed at the same time by the same polymerase under basically identical conditions. And so you have this what's called an operon structure. We have multiple different coding sequences which are going to end up coding for different proteins. So here is the case for the so-called trip operon, tryptophan operon. It's involved in making proteins which are important for making tryptophan. Here we make one big messenger RNA, and that gets translated into multiple different proteins. So this, as far as I'm concerned, is by far and away the most efficient way of doing this. Us eukaryotes are much less efficient. We've got genes, for instance, for tryptophan biosynthesis spread out all over the place in all kinds of different chromosomes. Each of these is transcribed separately, and then each of them is translated into protein separately. This is a much more complicated process. Allows a lot more regulation because you can regulate each of those genes, each of those messenger RNAs separately, but you have to have that extra level of regulation. Whereas in the bacteria, which are much more streamlined here, you can have one level of regulation for all of these things. Here in this slide, it mentions also RNA processing. And again, hopefully this is all review for you. Most of these eukaryotic genes are not just from start site of translation to stop site of translation. There are lots of these so-called introns. And it turns out that in most cases, introns are actually much longer than the exons or the protein coding sequences. However, when these pieces of DNA are transcribed, the template has both exons and introns in it. So you make this so-called primary transcript with lots of introns and exons in it. You put a 5' cap on the end, and again, we'll talk about that cap structure much more later on. You put on a poly A tail, then you chop out all of these introns, and finally you end up with a messenger RNA down here at the bottom, which can actually be translated. And again, we'll talk a lot more about these individual steps later on in the class. But now I want to switch gears and talk about translation. And again, this is all about this wonderful RNA machine, which is the ribosome. Ribosomes are really composed of two large RNAs, but they can't function without other RNAs, and those RNAs are, of course, your messenger RNA and your transfer RNAs. The messenger RNA has the information on it. All that information is split up into sets of threes, also known as the codons. So 3 nucleotides to a codon. Codons interact with anticodons on your tRNAs. Those anticodons on the tRNAs give you the specificity for the appropriate amino acid. So the code down here is read by the tRNA, gives you the right amino acid. And then it's the RNA in the ribosome which catalyzes peptide bond formation between the amino acids that are hooked onto each of these tRNAs. And so the ribosome, as they say, clunks along the messenger RNA. It's actually probably more the messenger RNA that moves through the ribosome. All these things are relative. And in each case, you have these tRNAs that are associating with it. Let's look at some of these individual steps in a little bit more detail. Again, codons, 3 nucleotides. 3 nucleotides are then going to give you one particular amino acid. How was this originally found out? Well, turned out it was discovered by people who were able to synthesize RNAs. And it was a lot easier to make RNAs that were all the same rather than RNAs with lots of different sequences. And again, this is synthesized chemically. And then if you look at an RNA which has just uracils in it, and just uracils is great because you know it's RNA and it's not DNA. So just uracils, if you put that into a mixture of ribosomes and tRNAs, you get a whole stretch of phenylalanines. And what that tells you is UUU is the code for phenylalanine. AAA is the code for lysine, CCC for proline, etc. So this is all in the experiments where people took synthesized RNA, threw them into an extract which had ribosomes and tRNAs in them, and they got these stretches of amino acids. So this is great, but it only gives you 3 amino acids, and it turns out poly G doesn't work, even though poly G would give you lysine, because poly Gs like to form really stable secondary structures. And secondary structures, when I talk about nucleotides, that's stem loops, base pairing with each other, et cetera. Turns out G is pretty good at pairing with G. So these didn't work in those particular cases. So, okay, gives you 4 of how many amino acids? 20 amino acids. So how are you going to get the other 16? Well, it turns out that if you make these alternating nucleotides, which is relatively easy to do, then you end up with half and half mixtures, UC, 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 which gives you 2 different amino acids. So here UCU codes for serine, CUC codes for leucine, etc. So you end up with a peptide that has half serine and half leucine. And you can do all of this mixing and matching, and you end up with a more or less complete table of the genetic code. There are a couple of tricks you have to do, and if you're interested in that, come take my molecular genetics course in the spring, how exactly that took place. So, trinucleotides. One of the things that people found, which was really quite interesting, and this was, again, very early on in the days of making RNAs and then looking at what you got out, they made these sequences that were, AUC, 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 AUC. And strangely enough, what happened was they ended up with multiple different peptides that came out, either all isoleucine or all serine or all histidine. And the reason for that is that, well, the genetic code starts at a particular position and then goes 3, 3, 3, 3, 3. And those 3s can then, of course, be encoding different things. And so that is the difference between some potential genetic codes that actually people thought were being used and what we now know is the actual genetic code. So hopefully this, again, should be review for all of you. No, I don't expect you to remember it, with 1 or maybe 2 exceptions. And that one exception is AUG. This is the start codon, but also the codon which encodes for methionines. First position A, second position U, third position G. There's also this box up here, starting with U in the first position, A or G in the second position, and A or G in the third position. These are the stop codons. So this is where you stop translation. One of those in this block of 4 is the tryptophan codon. And in some cases, we'll see there's some genetic codes which actually use these stop codons for some other things, usually tryptophan. So what we know about the genetic code, a couple of different things. One, and this comes from that data where you get the 3 different polyamino acids out from that repeating trinucleotide sequence, non-overlapping. So it's 3 nucleotides, 3 nucleotides, 3 nucleotides, not 1, 2, 3, 2, 3, 4, 4, 5, 6. There are no commas. It's 1, 2, 3, 4, 5, 6. It's not 1, 2, 3 gap, 5, 6, 7. Almost universal, and that's where I mentioned you've got some differences, usually in those stop codons, where some of them are going to be encoding for tryptophan and some of them are not. Highly degenerate. What I mean by the highly degenerate, one of the things that I forgot to emphasize here is that there are a number of amino acids that are encoded for by multiple different codons. Leucine is one of the extreme ones here. 6 different codons can code for leucine. Same thing is true for, let's see, glycine, a couple of other ones as well. But you can have either single, in the case of AUG, or up to 6, in the case of Lu. Many of them are 4. So basically, in this case, if you've got your first position is a U, second position is a C, that is by definition serine, because it doesn't matter which any of these 4 other ones are. Yes, you can also talk about it as being to some extent redundant, although it depends on how you define redundancy. But yes, no, it's more the degeneracy rather than the redundancy in this case. Because as we'll see, the way you read the genetic code with the tRNAs is then going to be dependent on what those actual nucleotides are. So it's not really a redundant case there. You can't get rid of individual codons. In fact, it's very hard to do. People have done that, though, and if you're interested, send me an email. I'll send you some of this totally cool synthetic biology, but that's a different story completely. But basically, no, you can't get rid of individual codons. So they're not really redundant in that sort of terminology. Unambiguous, again, if you've got AUG, it's going to be methionine, and what we say, UUU is going to be phenylalanine. So these are things which, again, with the exception of synthetic biology, which, again, is a totally different story. Those exceptions, again, are almost always going to be in the stop codons. UGA, which is one of the stop codons in some organisms and most mitochondria. Tryptophan, some cases you have a stop codon that's encoded, gives you a glutamine. Some gives you a cysteine. Then the actual details of this table are not critical, but there are some differences that are present in the genetic code. So it's not a completely universal case. Most of these are probably derived. And so you had the standard genetic code to start with, and then these organisms, for a number of different reasons, have moved away from that. Yeah. Okay, in that particular organism, and so these are different organisms. So you don't have, and again, it turns out because it has to do with the tRNAs and exactly which tRNAs you have under those conditions. And so the euploides, which almost nobody works with, and the reason we found a lot of these things has to do with genome sequencing. Did a whole bunch of genome sequencing, and you said, whoa, wait a minute, that's really bizarre. There shouldn't be all these stop codons here. And they went back and looked at some of them and said, oh, actually, it's not, in fact, a stop codon there. But yeah, it has to do with the tRNAs and the actual tRNAs that you have in that particular organism, or in the case of the mitochondria, it'd be a particular organelle that will have that particular tRNA that's associated with it. Okay, we talked about this already, but it bears repeating. That fact that you have 1, 2, 3, 4, 5, 6, 7, 8, 9 is what's called the reading frame. In reading frame 1, you're starting at nucleotide 1. Gives you one amino acid sequence. Reading frame 2 gives you a different amino acid sequence. Reading frame 3 gives you yet another amino acid sequence. In theory, all of these could be used. Some viruses do use them. Those who've been in virology know that. But in most cases, in most cellular organisms, you're just going to use one of those reading frames. You don't use the multiple overlapping reading frames. And probably the reason for that is if you have a mutation at one particular position, that's going to change 3 potential different proteins as opposed to one protein relative to all of the other ones. And mutations happen. We wouldn't be here if mutations didn't happen. So that's that process. Excuse me. So mutations, reading frames, how do you go from that language of nucleotides to the amino acid language? Well, to do this, of course, we now know that there are tRNAs that are in this process. Amazingly enough, it was Francis Crick who came up with this idea in the first place. He talked about this adaptor hypothesis, where he said, okay, there has to be some way of going from this nucleic acid sequence to the amino acid sequence, and he thought it was small RNAs that were involved in that process. But basically what he said is, well, there are 20 different amino acids, but there are a whole bunch more different codons, 64. If you take away 3, that gets you down to 61 actually coding codons. And so how do you go from 61 to 20? And basically what he said was, okay, well, you've got these amino acids, which are where you have your specificity, and then this adaptor molecule can actually be a little flexible in terms of how it actually interacts with that messenger RNA. So his prediction was that there were at least 20 different adapters, didn't call them transfer RNAs at that time, RNA molecules, and that these amino acids were actually hooked up to these adapters before they got put into translation. And just Francis Crick was just amazing in terms of understanding these things. Of course, we now know that this is what happens. Now, however, to do this, you have to have some way of recognizing this amino acid and this adapter in the absence of knowing what the code is down here. And so basically you have to have some way of pulling these adapters together with the correct amino acid. And so this is the job of the amino-acyl tRNA synthetases. And many people would argue, particularly Dr. John Perona in the chemistry department here at Portland State University, because he works on these things, amino-acyl tRNA synthetases. These are the really critical part of the genetic code. And we know that you can chemically change the amino acid at the end of a tRNA molecule to a different amino acid. The ribosome could care less. It doesn't matter. It'll put in whatever amino acid you have at the end of that tRNA. And so making sure that you get the right protein is really the job of these amino-acyl tRNA synthetases. And this is the blob model for how that works. You have a tRNA which is specific for phenylalanine. In its anticodon, which is base pairing with the codon, it has AAA. Doesn't the codon for phenylalanine UUU? Yeah, because it's base pairing to it here. So the base pairing interactions. So this is just your tRNA, which is specific for phenylalanine. Then you also have your amino-acyl tRNA synthetase that's specific for phenylalanine. It now links this amino acid to the tRNA through this ester bond. And so the amino-acylation, this AA part of amino-acyl tRNA synthetases, that's putting this appropriate amino acid onto this tRNA. And it turns out that these tRNAs, the way that these amino-acyl tRNA synthetases recognize these tRNAs is not so much the anticodon per se, but also a lot of the side chains that are present in each of those individual tRNAs as well. Once you have an amino-acyl tRNA, this now is the substrate for the ribosome. It binds to your messenger RNA. One other thing to bear in mind, this base pairing interaction that happens between tRNAs and messenger RNAs is also always going to be opposite polarity. So if you've got the messenger RNA here, 5' UUU, it's not a great example. Here, the tRNA is going to be the opposite orientation. So the 5' end of your tRNA is here, the 3' end is here. And so you're always going to have the anti-parallel interactions with the base pairing that happens in between. Amino-acyl tRNA synthetases have to pick the right amino acid, otherwise, again, you're going to have the wrong incorporation of your amino acid. And it turns out that amino-acyl tRNA synthetases are really good at making sure they put on that right amino acid by something that's called proofreading or editing. Here, if you have the amino-acyl tRNA synthetase, this big green blob here, which is added on a particular amino acid, sometimes it puts on the wrong amino acid. And this is quite easy to comprehend if you think about the structures of things like leucine or isoleucine. These differ only by a methyl group. They're extremely similar to each other. Some things are going to have slightly different length side chains, asparagine, glutamine, etc. So chemically, these things are very similar to each other. And so many of these amino-acyl tRNA synthetases will put on the wrong amino acid right here, but then they'll cut it back off if it's the wrong one. The right one can't fit into this particular editing site. If it can't fit into this editing site, then it stays on your tRNA and you know that it's actually the correct one. And this process actually gives you extremely high fidelity. It's on the order of 1 in about 50,000 of any given incorporations in translation that gives you the wrong amino acid which gets put in. And that's almost all coming from these amino-acyl tRNA synthetases. Yeah?
Sorry, on the previous slide where the amino acid binds to the tRNA, is that oxygen is bound to on the phosphate or on the, say, 2' hydroxyl?
So in this particular case, the one right here, this OH at the end, yeah, this OH is the 3' OH. Okay. So yeah, it's not the 2' OH that you have there. It's on the backbone, yeah. So it's not a side chain. This is actually attached to that backbone. Okay, other questions on that? So wanted to spend a couple minutes talking about the structure of tRNAs. Everyone draws tRNAs as these wonderful cloverleaf structures. That's, of course, not what they look like in the cell. They look like this, but this is a lot harder to actually look like. I say, think about all of the changes that you have here. So people will unfold these. Basically, these two loops on either side wrap around each other in order to give you this structure here in B on the right-hand side. A couple of things to bear in mind on these tRNA structures. The first one, as we've mentioned already, the codon anti-codon interactions. Unfortunately, this slide's flipped them around relative to each other. So the codon at the bottom goes from 5' to 3'. Here, GCG. Anti-codon, of course, opposite strands. 5' end of your tRNA continues along. First position here of your anti-codon is going to interact with the third position of your codon, second with the second, third with the first. A couple of things here to bear in mind. Heavily modified. All of these tRNAs have multiple non-standard nucleotides that are in them. Most of these nucleotides are changed after you make the RNA in the first place. So you transcribe your tRNAs, then there's multiple modifications that happen to them. These are just a few of those modifications here. Pseudouridine, shown as this psi, also known as the T-psi-G loop, because this is always a sequence which is always this structure right here. D-loop, the dihydrouridine group. In some cases, you have methylated guanines. In many cases, at the first position of your anticodon, you have inosines. But again, these are things which are being modified often much after you have the transcription. So they're post-transcriptional modifications that happen to tRNAs. Again, remind you here, some amino acids have up to 6 codons. Leucine, arginine, glycine has 4 codons. But it turns out, in most cases, you have one or a few tRNAs that interact with that. And you kind of know this if you look at amino-acyl tRNA synthetases. The amino-acyl tRNA synthetases, there's only going to be about 20 of them as opposed to 61 different ones. So how can this be working? Well, our friend Francis Crick came up with this hypothesis, also known as the Wobble hypothesis. And basically all that it says is one tRNA can bind to multiple different codons. How does that work? Basically, it's almost all about position 1 in your anticodon. We saw in that last, 2 slides ago, that very often that's an inosine. Why inosine? Because inosine can base pair with uracil, cytosine, or adenine. And so it gives you multiple different possibilities on how these guys can actually interact with each other. Turns out that at the first position in the anticodon, G normally, of course, is going to base pair with C, but it can also base pair with U. C's always G's, A's always U's, and U's A's or G's. What do these base pairs look like? These are the Wobble pairs. In fact, this is almost an identical copy of the picture from that 1966 paper by Francis Crick, where you have GU base pairs, CI base pairs, UI base pairs, etc. And so inosine, which in fact is a modified adenosine, can base pair to almost all of the other potential nucleotides that you can have on your messenger RNA. This is a bit more, getting into a little bit more detail. This is a different tRNA here. This is the alanine tRNA. In the case of the alanine tRNA, you actually have 77 nucleotides. 10 of those are modified. So it's a really huge percentage of your nucleotides that are modified in any given tRNA. These tRNAs are usually about 80 base pairs in length. There's one more thing that I wanted to mention here, and that is that there's this sequence at the 3' end of almost all, again, it's biology, there are always some exceptions here, has CCA, so cytosine, cytosine, adenine. Turns out that this is a really critical sequence because it binds to the active site in those amino-acyl tRNA synthetases. And so amino-acyl tRNA synthetases have to have CCA at the 3' end of all of these tRNAs in order to be able to modify them, put that amino acid onto the end there. And here at the bottom, they're always base paired. Basically, this is a reminder that all of these guys are, in fact, interacting with each other through Watson-Crick base pairing interactions with, of course, all these modifications, such as these I base pairs which happen. Show you what some of these modifications look like. Uridine to pseudouridine. Basically, what happens here is you take this base, the uridine base, and flip it over 90 degrees. So this nitrogen ends up over here. Your double bond is switched, and now you have a bond between your sugar and a carbon. There's a pseudouridine synthase which does this. Some of the other modified bases are methylguanine, dihydrouridine. Also, in some cases, you have sulfur. Here's our inosine, which is basically an adenine missing that NH group right here. And so this now allows it to base pair with multiple different nucleotides. Finish up with our last set, and this is only a portion of all those different modified nucleosides. If you're interested in how all of these function, go and talk to Dr. Royal over in the chemistry department, because that's exactly what his lab does, is trying to figure out a lot of the different structures that you have here. You've got methyladenosine, isopentyl groups that get added. One of my favorites down here is lysidine, which is a lysine group, which actually gets added to your nucleotide. And then probably one of the most famous of these is the so-called W amino acid, or yosine. We end up with yet another ring structure, which has been added to your base structure. So, of course, this is going to cause all kinds of changes in your structure, which of course is going to be important for the function of that particular nucleotide in your tRNA structure. So it's now 10:04, and we'll stop here. We'll talk about the ribosome next. Now, quick question before I finish. I would say you need to know position of every atom. No. That there are multiple modifications. Pseudouridine I would know, because it's something which is really common. And we talked about it quite a bit. Inosine I would look at. But above and beyond those, that there are a whole bunch of them, yes, I would know. Exactly what each of them are, I can't expect you to know that.